All right, the last part of cellular respiration is called oxidative phosphorylation. So, just think about that term. So already we're going to have an oxidation. Remember, oxidation is loss of electrons. And then later on, there's going to be a phosphorylation. That's going to be ADP is going to get phosphorylated to become ATP. So it's going from its low energy state to its high energy state of ATP. Okay. So after glycolysis in the citric acid cycle, we've produced NADH and FADH, and they're going to have most of the energy. And remember, these are holding on to electrons. I like to think of it as electron wallet. So they carry those electrons to what's called the electron transport chain, and that is embedded in the inner membrane of the mitochondria. So here's a diagram. All right. So up top here is the intermembrane space. Below here we have the matrix of the mitochondria, and there's a handful of proteins here. We call that the electron transport chain, and it's used to move electrons from one point to another. Okay, so we have NADH over on the left here, and it's going to get oxidized. It's going to lose its electrons, but those electrons have energy, so it's going to take the energy is actually going to be utilized to pump hydrogen from inside the matrix to the intermembrane space. So think about this. We talked about pumps previously. We usually talk about how ATP is used. Well, here's an instance where actually just electrons, the energy of electrons are utilized to pump hydrogen from the matrix into the intermembrane space. Well, those electrons move from this first protein over into this next pump here, where those electrons are utilized again to pump hydrogen into the intermembrane space. They go through something called cytochrome C, and then to this last pump, where hydrogen once again is pumped into that intermembrane space. All right. Ultimately, those electrons, though, that have gone from NADH all the way to this last pump here, ultimately they need a place to go. All right. So oxygen is what's called the electron acceptor. Oxygen will take those electrons from NADH and will incorporate it into a compound and make water. Okay. So think about that original equation. Where we have like sugar, oxygen, we make carbon dioxide and water. Well, we already used up the sugar. We already made the carbon dioxide. So here we're utilizing oxygen and we're making water. So the only reason we really need oxygen in this process is to have that electron acceptor. All right, those electrons get accepted by the oxygen. They make water. They have a place to go. Otherwise, if there's no oxygen there to meet up with it, there's kind of a log jam. The electrons won't be able to go any further, and if it backs up, then we're not able to pump hydrogen, and you'll see why that's important in a second. Okay, so we've gathered, we've created a concentration gradient of hydrogen, where we've got high concentration of hydrogen in the intermembrane space and a low concentration of hydrogen in the matrix. Okay, so what's going to happen is that hydrogen is going to diffuse through this enzyme right here called ATP synthase. Sometimes it's called ATPase. So when I was in school, it used to be called ATP synthetase. Either way, it's still the same protein. It still does the same thing. Okay, so those hydrogens diffuse through ATP synthase, and then kind of the kinetic energy from the hydrogen ions is utilized by this protein to help fuse phosphate to ADP to make ATP. All right, and then basically that's just it, right? So this is why it's a problem if we make a log jam with our electron transport chain. If we can't pump hydrogen through here, the hydrogen can't diffuse through the ATP synthase, and then we can't make any more ATP. All right, so here's kind of a close up of our ATP synthase. We're just it's kind of a model as to like what could be going on with it. And then here's just some text that is kind of just referring to what's going on. But we do call this this process chemosmosis, and this is going to make the majority of ATP in the cell. All right, so here you can see, just kind of giving you a rundown. By the way, oops, sorry, I forgot to mention this. So NADH starts all the way at the end, but FADH starts kind of one protein like downstream from that first pump. So FADH just pumps less hydrogen through, so it's not able to make as much ATP as, say, NADH. In theory, you can make 3 ATPs for every NADH and 2 ATPs for every FADH. There's going to be some efficiency problems in it, so it's not 100% like 3 and 2, but I know it's close enough. Anyway, so the max that you can make per glucose is we got about 36 or 38. It's very it's very precise number, but here we make 6 ATPs. There's 12. There's 6, 12. There's 18. And then, 24 was 18. Oh, wait. Yeah, anyway, it's a lot. That's like, I kind of lost track. Sorry. But ultimately, we make a lot from sugar, but it's mostly through oxidative phosphorylation. One of the kind of interesting things is that we kind of focus on glucose in cellular respiration. And I just want to take a quick moment to say it's, this is already complex enough, but, here's a kind of a simple view of how complicated this is, because as you know, we don't necessarily just use like monosaccharides as our kind of energy molecule. Sometimes we have triglycerides, and, the glycerol from triglycerides, that actually would kind of enter into glycolysis, but the fatty acids enter in that intermediate step between glycolysis and citric acid. Some sugars might enter in the midway through glycolysis. Amino acids might kind of get utilized towards the end of glycolysis, or that intermediate step, or kind of input themselves in the citric acid cycle. So basically, think of kind of our treatise on cellular respiration is just really kind of an introduction, and there's really a lot more going on. So thanks.